And now we can talk with Marion Koopmans. So Marion Koopmans is head of Erasmus Medical Center Department of Viruscience, and she will talk about lessons from COVID-19 for future disease, and it is also linked to preparedness. And as you did for some of you, and thank you really for asking your question, as you did for Frédéric Kirk, don't hesitate during the presentation. As soon as you have a question, don't hesitate to write them down into the window, the question window. So now I give the floor to Marion Koopmans. Thanks for participating.
Yes, thank you very much also for the invitation, and congratulations on your One Health Hub. This is, of course, a topic very near to my heart and areas of interest. So I'm happy to be able to share some views here. So what I have done is taken the COVID pandemic as an example of the importance of One Health collaborative work. And this is, of course, not coming new, although the pandemic was a surprise, maybe by many in the human health sector. This is not the case if you look at that from a virology perspective or from a One Health perspective. And there's already been the warnings, for instance, by Mike Ryan, the WHO Health Emergencies Director, who warned starkly for the rising probability of new disease outbreaks coming from spillover events, and particularly after the Ebola outbreak in West Africa. Now, if we focus on COVID-19, there is some interesting observations at the root of this as well. This is a publication that has reconstructed the trends in the pork prices in China, and has found a steep increase of those prices in the second half of 2019, and that was related to an epizootic, a major animal disease outbreak caused by African swine fever, which has hit very hard in China's big pork production sector, and has led to the massive culling of many pork, and as a consequence, these rising prices. So the authors here postulate that this is a classical example of the type of drivers that could lead to shifting meat consumption, for instance, wild animal meat, and thereby increasing the risk of a spillover event. Not proven, but interesting and thought-provoking observation. Of course, we have heard first about this a new disease from a notification here in a Chinese financial journal that had picked up a leaflet from the Wuhan Municipal Health Service mentioning that a cluster of cases, human cases with unexplained pneumonia and linked to a market, the Huanan seafood market. And my first thinking was, well, could this be a cluster of avian influenza? Because as the previous speaker mentioned, we are well familiar with the problems with avian influenza on live animal markets, documented in China, but also in other parts of the world. And here illustrated in data from environmental sampling, where there's swabs collected on stalls, on tables, on the animal market, and where at any time you can have up to 50% of those swabs positive, testing positive for avian influenza, including avian influenza of the subtypes where we worry about potential for human infections, H7 in red and H5 in blue. And these trends are influenced by animal vaccination programs that you see here, introduction of H7 vaccination and reduced presence of H7 viruses, but also H5 vaccination, where we see that reduction doesn't take place, and that's because of virus evolution, because that's the other concern, and that's what we see in those market systems, is just how much evolution and genetic evolution of these viruses there is. It's pretty much a virus zoo ongoing. This is now pointing at a part of China, but with the recent huge change in the ecology of avian influenza in wild birds across Europe and now in the Americas, we may start to see some of this in our own backyards as well. Back to SARS-CoV-2, the first detections can really be attributed to these persons. The clinician who treated the first recognized cases with this unusual type of pneumonia end of December, or 26, 27 December, and who picked up the phone and brought in the public health services and also sent samples out to different laboratories, including the laboratory of Professor Zhang, who with metagenomics pulled out a new virus and very importantly shared that information early on with the world in January 10th, I think, through virological.org. So very important steps in early detection of a problem. And here is information from the early paper, which really has been quite informative in my view, and an excellent example of how these types of outbreak investigations should be done. So it showed presence of the virus in acute phase samples from patients, not in later stage samples. It showed evidence of the virus in cell culture, evidence of an immune response related to the disease syndrome, and genetic information on the viruses, placing that into, showing it was really a novel pathogen for humans, but showing its relatedness to bat viruses. And the early stages, of course, were typical of what you see in early emerging disease outbreaks, with the first notified cases here. Then a time where there's uncertainty. If you ask around in winter, there will be many people with unexplained pneumonia until the course was clear. Diagnostic test kits could be made, and then the testing showed an increasing number of cases, obviously. Questions were about, has this maybe been simmering for a much longer time? And this has been one of the topics of the studies done by a mission that was put together to study the origins of the pandemic. And here's a piece of information that was pulled out, and that's looking at excess mortality in the city of Wuhan. That's shown here in gray. And seeing that the signal really starts the second half of January, and then there was a big spike in excess deaths. And this reflects clearly more widespread circulation throughout December, but not really much earlier than that. This is also confirmed by early genetic data, looking at the genomes of viruses from the earliest cases recognized by the public health investigation. And if you look at that, you see that there's already diversity, genetic diversity there, not a whole lot, but enough to conclude that these were not the first cases, so that there have been chains of transmission missed, which again is not unusual, certainly if you have a disease that has such a mild presentation in many people as well. The timing through molecular clock analysis put the time of estimated ancestor back to somewhere mid-November, towards the second half of November, so not a whole lot earlier. And that's also confirmed if you take sequence data from later time point and add in genetic data from a wider range of places. That picture is consistent, and there's been several publications about that now. Now, of course, when seeing a new virus pop up like that, it is, given what we know now, the immediate reflex, of course, is to think of an animal origin. And certainly for coronaviruses, there's a very good foundation for that. If you look at the evidence now for an animal origin for all of the coronaviruses that we know that can infect humans, with original reservoir hosts in bats and rodents, and with examples of spillover hosts for several of them. And of course, the question is what exactly has been the situation for this new virus, and that's still going on. But if you look at the initial genetic information and place that in the taxonomic tree of beta coronaviruses, those are listed here in pink. It is clear here is the SARS-like coronaviruses, here is SARS virus, and the new virus is also clustering within this scheme along in the middle of all other bat coronavirus. So of all the global virome that is known, these are the closest relatives, but there's still several decades of evolutionary time between the most similar bat virus and the human viruses. So there's a piece missing there. Looking at what exactly happened at the root of this pandemic, one of the investigations has been focusing on the Huanan seafood market, which had been mentioned as the most likely place of infection for some of the first recognized cases. And that does not necessarily mean that it happened there, because as you know, it's clear that the initial cases have been missed, but it also doesn't rule it out. And just looking at where cases occurred on the map of the market, it's a huge market with two different areas. Clearly, majority of cases mapped to the west area, which is, if you think of widespread community circulation, doesn't make sense. So it does suggest a link to the market. But if you then look at what was sold at the market, there was not really a link to any of the potential products that could be a source of a virus. Of course, most interest was in wild animals, and at the end of December, when this mapping was done, the people that did the study said there were no live animals. There was, of course, debate about that, but there was no clear documentation of that. But since then, there's been a publication earlier this year that found, or that really did surveys of several of the markets in Wuhan, and has good documentation of the presence of many different types of susceptible live wild mammals, including on this market up until end of November 2019. So that there is definitely a potential link. Now, question there is what types of animals to think of. And I think this is where SARS-CoV-2 has continued to surprise us, with an increasing number of animals shown to be susceptible. Here's an example: pangolin, a species that you would not immediately have thought of, but that does, if you really dive into a little bit of the ecology and epidemiology, it again has all the marks of risk for disease emergence, because these animals are critically endangered, but also are popular food. And because of that combination, they need to be brought in from an increasingly wide geographic range. And that's done through narrow bottlenecks, which are hubs that collect them and ship them on. In this case here in Vietnam. So that is, if you think about animal movements, concentrations, and viruses in that system, it is really favoring viral transmission. So there's been batches of pangolin tested on 2 separate occasions. They were historic batches seized by import authorities, and they harbored SARS-like coronaviruses, distinct again from the new virus, but quite closely related. So they are a potential reservoir as well. There's ongoing studies, of course, to explore, and that exploration is challenging because, of course, the potential reservoir is huge and very wide and geographically dispersed and doesn't stick to borders. And here's an interesting study that has looked at the presence of SARS-like viruses in Laos and found several highly similar SARS-related coronaviruses again in those bats, including viruses that have the receptor binding domain, so the part of the virus to which binds to the human host that is very similar to that of SARS-CoV-2. And there's also evidence that these viruses can bind to human cells. Okay, back to the early phase of the human outbreak investigation. So this is again the first month of the pandemic, where we started to see exports of cases. And here a first estimate from Imperial College based on statistical analysis of likelihood of having these exports given the travel patterns. They already said there must be thousands of cases by now. So a lot is going under the radar. And that's, again, something that is fairly typical of early emerging disease outbreaks, particularly when the case fatality rate is not that high. If you have an Ebola type situation where every other patient may die, that is, in fact, somewhat arguably, but it may be easier to track than if you have a disease with a profile of COVID, where the majority of cases will have mild or no symptoms. And then certainly if there's no diagnostics available and those symptoms in many people are sort of so non-specific, it is really difficult to understand what the size of the pyramid is. So how much is flying under the radar? And this is something that we've also learned from how Europe was seeded with this virus. Of course, the first after Wuhan and seeding into taking off of the pandemic in, for instance, Iran, northern Italy was at the core of the European pandemic, with the closing of 2 towns end of February because of an overburdened healthcare system, which reflected, of course, widespread circulation that had been ongoing and undetected for a couple of weeks already. And this was really right into the period where there was a lot of human travel, with winter sports very popular also for Northern Europe, and Carnival, which is also a winter fest that is popular in parts of Europe. So perfect dissemination and amplification setups in humans. And that's also reflected if we look at the virus side. So this is a genetic tree now of genomes of viruses from patients in our country. And this is in early March, 10 days after the first recognized human case, hospitalized case, when we did a screening of healthcare workers in 10 hospitals in a region that had this Carnival fest, where it's normal after Carnival you have respiratory disease. But this turned out to be already quite a lot of SARS-CoV-2 cases. And as you can see, they were already all over the place, reflecting multiple introductions and dissemination events, with here the central 2 clusters again that you saw in the earlier slides. So a virus that very much can go under the radar. Mark the date here. 10 days after the first recognized human case. That was the late November. Now, in the months following that, we have seen also animal disease situations pop up. So already in February, there was mention of infections of dogs and cats from China and Hong Kong. And that alerted a zoonosis meeting in our country where we discussed which animal species could potentially be susceptible. There was already indications that the most common livestock species may be less at risk, but mink came up as a potential high-risk species also because of its relatedness with ferrets, which were being used as an animal model at that time. So that really started a screening program for the testing of mink in an animal health surveillance program where dead mink are sent for autopsies and testing just to monitor what may be going on. And that popped up infection of these animals with SARS-CoV-2. And here is a summary of the genetic information, where in red you see human genomic data. In blue, you see the mink virus sequences. And what you can see is that there were 5 separate introductions of viruses from humans back into mink, and then transmission among mink, and from mink farm to mink farm, and from mink back to humans. You can see that most clearly here. So you have human infection, transmission to mink. These are several different mink farms, and their animal to human transmission again. And we've spent a lot of time to try and understand how exactly what exactly was going on. The initial attempt was to use the animal control, disease control measures that we know. But that turned out to be very difficult, and it was virtually impossible to control the spread of this infection. Here's a timeline. The colors here are those different clusters, and you see in red is the biggest cluster, which also persisted. It continued to spread. And what you can see is that there are activities, time periods where a lot of animal handling is going on, and then time periods where it's quiet. And from the phylogeny, it was possible to see that the introductions really happened in a short period of time, already early in the pandemic, possibly even before the first human cases were recognized. So in hindsight, all the human disease alert should have been paralleled with an animal disease alert. And here you can see what the time line was between the first detection of the problem in animals and the estimated time of introduction. If you have that type of situation, you can also try to understand from the genomic data coupled with other data to how exactly the transmission went. And you can see here a complex pattern of spread. Each dot here is a farm representing the 5 different clusters, where in some instances there was a clear link, for instance, workers that worked on both facilities, but in many instances there wasn't. We did find evidence of infection in escaped wild mink, which is a problem I wasn't aware of, but that apparently exists in many countries that have mink farming. So just from the sequences, it was possible to see where those mink got the infection. There was also evidence for infection in cats and in dogs and in feral cats that hang out near these kinds of farms and just get free food. The situation, of course, has been observed in many parts of the world, mapped here. Of course, we have important gaps in surveillance and knowledge. and with different animal species, mostly carnivores, so cats, dogs, mustelids, but also primates and several species of wild animals, and most recently the hamsters picked up in Hong Kong, but exported from Europe through our country. Again, another complex animal scheme. This is the recent observations, or not so recent anymore, from North America, where they have seen reintroduction spill back of this virus into deer populations. And again, with genomic data, you can see just how, well, messy the situation is, with at least 6 separate introductions into the wild deer population in just one state. And that was linked to hunting season, although I still have a hard time understanding how actually that is happening. Is that because maybe there is feeding to attract the deer for the hunters with contaminated food that introduced it? Is it human droppings? Well, I don't think we understand exactly how that works. What is a concern here? Well, the concern is that we do even in single animal passage, and this is data from experimentally infected cats, dogs, and hamsters, you do see evidence for selection on the virus. You see evidence for positive selection, which may be selecting against human risk, if you think about animal adaptation, but that is certainly not a given. And that selection is particularly observed in the spike protein gene, which of course is a crucial gene, because it is the part of the virus to which it uses to dock onto host cells, and it's also the key target for all the vaccines that have been developed so far. What we have seen in, for instance, the mink situation here from the Netherlands, is that we see these viruses evolve. I'm sorry, evolve in the animals and pick up mutations in critical sites on the spike protein. And here's a model of the spike protein. Here is one key domain that is targeted by neutralizing antibodies, and it's the part where the virus binds to the host cells. And as you can see, there are mutations that have been observed in humans and in mink. So there's co-selection for mutations that increase the risk of changes of the affinity of the virus, and also immune escape. And the same is seen in another important epitope right here. And what can happen then is, we've learned, and you're all familiar with, is the emergence of variants. Here's the older ones, alpha, beta, gamma, delta, and mu, and we're now at Omicron. And what we have seen is that with these variants, we have seen an increase in transmissibility. This is the famous R parameter. We've seen an increase in transmissibility and an increasing challenge in trying to contain this virus. So far, these viruses were selected for transmissibility. With Omicron, the key selection feature is immune escape, not so much another step up of transmissibility because of, for instance, higher viral loads. It's a different mechanism here. One issue coming up with Omicron is also the question, where does this happen? Omicron really is very different from the previous viruses that's shown in this compressed visualization from NextStrain based on the globally shared data. Well, here you have all the other variants, and here sits Omicron. And the big question is really, where did this happen? What made this virus so different from all the previous viruses? Is that a new introduction? Maybe. Has it been evolution within immunocompromised hosts? That is a popular hypothesis, because there is some evidence for that people with impaired immunity that cannot clear the infection accumulate mutations in epitopes. That has been observed, and the emergence of Omicron in African contexts raises questions about untreated HIV patients as a potential risk. And another hypothesis is maybe animal passage, and there has been mention of some unique mutations that have been observed in mice passaged viruses, but there's no real definitive proof for any of that. What I do think is this does point at so we are really sort of not at all in control of what is spreading and what is moving back and forth between the human and animal space. So I think in a true One Health vision that should be shared. So very much agree with the perspective from Frederick Keck that we should really move away from this human-centric thinking and consider human health, animal health, environmental health in combined, and that ecosystems that are shared with healthy and not so healthy pathogens, viruses and microbes, which we can detect through common and generic toolbox like metagenomics. So we have a common language in the form of DNA and RNA. And of course, it's very important to realize that we are scratching the surface in what we think is out there. This is what we know, and there's so much more to be explored, and it's just part of also in part the healthy ecosystems. But if you think in terms of preparedness, then a key way forward, in my view, and that's also what we're doing with the European consortium, is to try to address this question: can we use some intelligent design of surveillance to be earlier, to move away from this current situation where we really start to act when people get sick, whereas we know that many of the emerging diseases come from animals, livestock and wild animals, where there is their own disease dynamics and healthy and unhealthy situations. And can we maybe use that knowledge to predict where risks are increasing and then go with targeted surveillance? There's also questions, of course, about interventions at the animal side. This is an image that I liked from a paper that discusses that, and that argues for maybe considering also animal vaccination, not in the original wildlife reservoirs, but maybe in these animal species that live closer to humans. And there certainly currently is vaccination studies ongoing in mink. Again, then from a human health perspective, I think that needs to be monitored with caution because of this co-selection for possible drift variants situation. So you don't want that going under the radar either. So with that, these are sort of my key messages. I think this pandemic just shows how critically important it is to really study emerging disease outbreaks jointly from the human side, animal side, environmental side. The widespread spillback of SARS-CoV-2 raises concerns about generation of new animal reservoirs with continued circulation, a bit like what we have been seeing with pandemics of flu and swine influenza. Animal impact of SARS-CoV-2 on animals can be high. So mink farming has been stopped in the Netherlands, in Denmark, and has suffered heavily in some other parts of the world. So depending on how you view those kinds of developments, there's certainly very high impact. And we have the risks of virus passage, co-selection of variants that increase the risk. And future preparedness, I really think, needs to consider this One Health approach. And with that, I would just really want to acknowledge funding, also including projects in place that are already were in place to work on preparedness, and that I think we need to go back to rethink and strengthen in the near future.
Thank you very much, Marion Koopmans, for this passionate, really exciting presentation, which was so deep into the topic and which helps us understand much better what was at stake with COVID-19 and what is maybe about to happen again. So I have several questions for you, and I remind everyone of the opportunity to ask your questions in the questions window normally, but the bottom right of your screen. I see that a few questions are already coming. So thanks. Just before asking the questions from the audience, I have a really important one for you to start. Do we know to which extent the SARS-CoV-2 virus was before its emergence in human, particularly prone to cross-species barriers? And would an increase in spillback transmission lead to an exponential increase in the risk of re-emergence of variant for humans, which is really in the core of the questions today?
Yeah. So looking at the early stages, so the molecular clock dating really puts the time of origin from very different estimates by many different groups, not so far before December 2019. So something may have happened there. Given what we know now about the range of animal species that is susceptible, there's quite a bit of choice. Of course, looking at the sequences, bats are a very likely original reservoir, but whether there has been a bridging host, we don't know. There's been quite extensive testing of animal species in China, more than 80,000 samples processed, and that hasn't found a smoking gun. But there's also some species really lacking from that list, which includes mink, which includes prairie dogs or raccoon dogs, for instance, which are a big product in markets in China. So I think that's question is unsettled. As for the risk of spillback, I do think that may come back to haunt us, because what we do see is continued circulation. We've seen in mink several the reason why we can tell people are infected is because of with circulation of these viruses in animals, you get a clear signature. You can recognize these are viruses that have been passaged through animals, and we see evidence of adaptation, which is an unpredictable process. So what is possible, in my view, is that we would see continued divergent evolution of the virus in humans and in animals, so that you get problems with possibly antigenically distant viruses spilling back again from a new reservoir animal. So I certainly think that really requires attention.
So many questions about these matters and these variants and the going back from wild animals. And I've got one which is, of course, in everyone's minds from Cédric Poissonnier. What are the odds of having another mutation that affects a large number of human populations, such as lately with Omicron, of course?
Well, the odds are high. There is now almost a synchronous global wave of Omicron, which, of course, will, given how big that is, will give also a bit of an immunity wall. But we've learned that immunity is certainly not sterilizing. And as long as there's so widespread circulation, there's certainly also an opportunity for selection of variants. Omicron is the first one where there's very strong immune selection, and it's quite likely that process will continue. And what is also a concern is that so there's big differences in the world in background. So Omicron infects now people as primary infections and people that have been previously exposed or vaccinated. In the second group, you see quite a broad immune response, cross variants. In the first group, that is more narrow. So if you would think of variant emergence back from Delta or an earlier virus, it is quite likely that Omicron infected primary infected group would still be quite susceptible to those, as we've seen the other way around now. So there's concerns like that, and that's also translated into, there's a discussion now whether the vaccines should be updated for Omicron, and there's work ongoing. But because of this, it may be wiser to look for bivalent vaccines. So have Omicron plus some of the older variants that really are antigenically tighter.
Okay, so it leads to the general matter and question of vaccination. According to you, which is so sensitive question in France, namely, and in many other countries anyway, is there, according to you, any real impact or strong impact of the vaccination strategy on the evolution of the virus itself?
Of course, this is always a trade-off, and we know that. We know that viruses continue to evolve. So as long as you have replication and you have some kind of barrier or bottleneck, you run the risk of selecting for viruses that escape that bottleneck. And that's, of course, also what happens if you talk about immune escape. But what we do see is that, for instance, even with Omicron, where there's clearly escape from antibody immunity, neutralizing antibody immunity, the protection from disease, which is T-cell based more, remains quite high. So this is a complex story. So yes, there is immune selection. The more immunity there is from natural immunity, infection immunity, and from vaccine immunity. And that's something we will have to deal with in moving forward.
I have a more technical question, which is related to other points that you talked about in the presentation regarding the potential relationship between COVID-19 and African swine fever you presented. Is there concerted effort to study connected co-occurring emergencies?
Not to my knowledge, but that's something that's so in I'm involved, or I'm coordinating a European effort. It's called FIO. It's a project where we're trying to use that kind of thinking. So if you see examples like this, and if you look at outbreaks of emerging diseases, very often there is, if you look back, there are some drivers where you think, okay, something changed very dramatically here that could have led or contributed to this emergence. So what we're thinking is maybe if you start collecting information about drivers of disease emergence, big changes in the animal sector, clearing of forests, things like that, and you have a data intelligence hub that tells you, hey, something is really changing here, like the big culling of pigs in because of African swine fever. Think of what that does to the balance between humans, animals, and environment, because that's what typically creates a high risk situation for a spillover. And then that could also target your surveillance. That's the line of thinking that we're trying to develop.
All right. Go back to the SARS-CoV-2, because there's so many questions about it. Do we know to which extent the SARS-CoV-2 virus was mutating and changing within animals before its emergence in human? And is that only a number game, increased transmission, or human are a good host for mutation? How can we prepare for this increased diversity and its impact to control and vaccination?
I think that's 5 questions or something like that.
Sorry,
So,
sorry.
so if you look in the reservoir, there's been quite excellent studies looking at the diversity of SARS-related coronaviruses in bats, for instance, and there's very clear evidence for quite substantial recombination in the evolutionary history of those viruses. And there's also the work of EcoHealth Alliance has linked that to also environmental niches where there is, for instance, cave systems that really harbor multiple species of bats. So that those ecological niches is where they also find quite a lot of evidence for recombinant viruses. So that's certainly ongoing, and it's of course a bit of a, well, I mentioned the virus Su in the market system in southern China, but that's certainly also happening in wildlife. And what I think what we've seen in humans is a host adaptation. So there was the initial discussion, this virus is already fully human adapted. Yes, it was able to infect, but already early, so with the dissemination into Europe, so February 2020, there already was a critical mutation that really increased the level of replication of this virus in humans, that really increased the transmissibility. And then every variant since then has been a step up of that transmissibility. So you can see that I look at that as part of host adaptation process, which is continuing. And of course, it will at some point reach some kind of equilibrium, we all expect. But when exactly that will be, not sure yet.
Okay, we'll see.
And what is interesting in relation to the animal host is that with these evolving human variants, you have a different pattern of animal host susceptibility. So the first viruses were not able to infect mice until alpha virus variant. And then mice became infectable. So there's also changes in the animal host range with new variants. It's a very complex picture.
Yeah, very complex, is what we can see. A last question, we've got 1-2 minutes left. So looking back at mortality data that illustrated the first sanitary effects of COVID-19 in the Wuhan region, how to deal with previous outward mortality detections that do not seem to be related to a future pandemic, as there is a risk that many cases might fly under the radar. There is probably a critical trade-off between sensibility and specificity.
Yeah, so I think that's one of the key challenges in early warning surveillance is how do you pick up those very small clusters, but maybe the warning signs, and is that even possible? Of course, that's also where something like serology comes in, because of course, when hunting for the virus, to use Frederik's term, you only have a very narrow window of detection. If you include serology, say serology in food animal handlers, you have you would do that repeatedly, you may have a better way of monitoring what is going on, even if there's no overt disease in these people. So that's part of the risk-based surveillance concept. It never would be prevention, but it would be early detection. The real question then is, can you be really moving to prevention, which means looking for changes in the ecology in animal world? And of course, that's extremely difficult. But yeah, I think we should consider it.
Yeah, indeed. Thank you very much, Marion, for this so exciting and precise and deep presentation about this topic that concerns everyone. Really, thanks a lot for your participation.